Bacterial cell structure. Flagella and their fractions. These are extremely thin hair-like appendages. They come out through cell wall and originate from basal body, structure just beneath the cell membrane in the cytoplasm. They are made up of protein flagellin. On the basis of presence of flagella, pattern of arrangement of flagella, and the number of flagella present, bacteria are classified into different taxonomic groups. Atricus means bacteria are without any flagella. When single polar flagellum is present, then the condition is known as monotrichous. If tuft of flagella is present only at the one pole of bacteria, then these are lophotrichous. Amphitrichous is a condition when tuft of flagella at each of two poles is present. In peritrichous form, flagella surrounds the whole cell. Most of the bacilli and spiral-shaped bacteria have flagella. Cocci very rarely have flagella. Primary function of flagella is to help in motility. With the help of flagella, flagellate bacteria can also detect and move in response to chemical signals, which is a type of behavior called as chemotaxis. Villi and their functions. These are hollow, non-helical, filamentous appendages. True villi are only present on the gram-negative bacteria. They are made up of special proteins called pilin. They are primarily involved in mating process between cells called conjugation process. Some villi functions as means of attachment of bacteria to various surfaces. The cell envelope, the outer wrapping of bacteria. Bacterial surface and walls are very diverse. Collectively, complexes of the layer external to the cell protoplasm are called as cell envelope and include capsule, slime, and cell wall. Capsule. Bacteria produce capsule, which is made up of repeating polysaccharide units and of protein, or of both. Capsule is tightly bound to the cell. It has thicker gummy nature that gives sticky characters to colonies of encapsulated bacteria. It provides pathogenicity. Slime. Some bacteria are covered with loose, soluble shield of macromolecules, which is called as slime capsule. And slime provides greater pathogenicity to bacteria and protects them against phagocytosis. Cell wall. Beneath the extracellular substances and external to the cytoplasmic membrane, cell wall is present. It is a rigid structure. It determines the shape of bacterium. Cell wall also protects the cells from osmotic lysis. Cell wall is only absent in mycoplasmas. Christian Graham developed the technique of Gram strain. Bacteria could be divided into two groups based on their response to Gram staining procedure. Gram-positive bacteria are stained purple, retain the primary dye due to the formation of CV1 complex. and gram-negative bacteria are stained pink, retain secondary dye in the color. There are many structural differences between the two groups, which are the primary basis for the difference in the staining behavior. The cell walls of most bacteria have a unique macromolecule called peptidoglycan. Its amount varies in different types of bacteria. It's composed of framework of long glycan chains, cross-linked with the peptide fragments. The intact cell wall also contains chemical constituents such as sugar molecules, teichoic acid, lipoproteins, and lipopolysaccharides, which are linked to peptidoglycan. Several bacterial groups lack the cell wall structure, characteristic of gram-positive or gram-negative bacteria, and some bacteria have no cell wall at all. Cell walls of archaebacteria are different from eubacteria. They do not contain peptidoglycan. Their cell walls are composed of proteins, glycoproteins, and polysaccharides. Just beneath the cell wall is the cell membrane or plasma membrane. It's very thin, flexible, and completely surrounds the cytoplasm. Plasma membrane is very delicate in nature. Any damage to it results in the death of the organism. Bacterial membranes differ from eukaryotic membranes in lacking sterols such as cholesterol. Cell membrane regulates the transport of proteins, nutrients, sugar, and electrons or other metabolites. The plasma membrane of bacteria also contains enzymes for respiratory metabolism. Cytoplasmic matrix. The cytoplasm of prokaryotic cells lack membrane-bound organelles and cytoskeleton, like microtubules. The cytoplasmic matrix is the substance present between the plasma membrane and the nucleoid. It has gel-like consistency. Small molecules can move through it rapidly. The plasma membrane and everything present within it is known as protoplast. Thus, the cytoplasmic matrix is a major part of the protoplast. Other large discrete structures such as chromatin, nuclear body, ribosomes, mesosomes, and granules and nucleoid are present in this matrix. Nucleoid. A bacterial cell, unlike the cells of eukaryotic organisms, lack discrete chromosomes and nuclear membrane. The nuclear material or DNA in the cell occupies a position near to the center of the cell. This material is single, circular, and double-stranded DNA molecule. It aggregates as an irregular shaped dense area called nucleoid. This chromatin body is actually an extremely long molecule of DNA that is tightly folded so as to fit inside the cell component. Since bacteria have a single chromosome, they are haploid. Other names of nucleoid are nuclear body, chromatin body, and nuclear region. It's visible in the light microscope after staining with the fulgin stain. E. coli closed circle chromosome measures approximately 14,000 micrometer. Plasmid. Many bacteria contain plasmid in addition to chromosomes. They are circular, double-stranded DNA molecules. They are self-replicating and are not essential for bacterial growth and metabolism. They often contain drug-resistant, heavy metals, disease, and insect-resistant genes on them. Plasmids are important vectors in modern genetic engineering techniques. Ribosomes. Ribosomes are composed of RNA and proteins. Some may also be loosely attached to plasma membrane. They are protein factories. There are thousands of ribosomes in each healthy growing cell. They are smaller than eukaryotic ribosomes. Mesosomes. The cell membrane invaginates into cytoplasm forming structure called as mesosome. Mesosomes are in the form of vesicles, tubules, or lamellae. Mesosomes are involved in DNA replication and cell division, whereas some mesosomes are also involved in export of extracellular enzymes. Respiratory enzymes are also present on the mesosomes. Granules and storage bodies. Since bacteria exist in a very competitive environment where nutrients are in short supply, they tend to store extracellular nutrients when possible. These may be glycogen, sulfur, fat, and phosphate. In addition, cells contain waste materials that are subsequently excreted. For example, common waste materials are alcohol, lactic acid, and acetic acid. Spores. Certain species of bacteria produce spores, either external to the vegetative cells, exospores, or within the vegetative cells, endospores. They are metabolically dormant bodies and are produced at late stages of cell growth. Spores are resistant to adverse physical environmental conditions such as light, high temperature, desiccation, pH, and chemical agents. Under favorable conditions, they germinate and form vegetative cells. Cysts. Cysts are dormant, thick-walled, desiccation-resistant forms and develop during differentiation of vegetative cells, which can germinate under suitable conditions. They are not heat resistant.